Good morning and hello everybody. My name is Emma Doyle. I'm pleased to welcome you here today to join us for this online workshop on Caribbean coral rescue in the face of stony coral tissue loss disease. This workshop has been convened by the Caribbean Cooperation Team of Florida's Stony Coral Tissue Loss Disease Response Effort. It's being jointly hosted by NOAA's Coral Reef Conservation Program, AGRA, the Atlantic and Gulf Rapid Reef Assessment Program, and MPAConnect through GCFI. Now, the material today will be presented in English or in Spanish, and we are offering simultaneous interpretation of all of the oral presentations into English, Spanish, and French. So please select your preferred language at the bottom of your screen. You can change your selected language at any time. Now, today we have some presentations for you and we have some time for questions, as always. During the session, please type your questions into the chat box, preferably in Spanish or in English, please, and we'll facilitate a question and answer session after we've had the first two presentations, and then we'll facilitate some more questions and answers at the end of the last of our other country presentations. So please also feel free to type any greetings or comments into the chat box for everyone. These won't be translated. Now, due to the large number of participants who we anticipate today, everybody is currently in listen-only mode. So please make sure to use the chat box to communicate. Now, first up, I would like to introduce Dana Wizinick-Mendez, who is our Atlantic and Caribbean team lead with NOAA's Coral Reef Conservation Program. She's based in West Palm Beach, Florida, and has been working to lead and support the efforts to respond to stony coral tissue loss disease in Florida and the Caribbean since the outbreak was first detected. Dana and Judy Lang with AGRA co-lead the Caribbean Cooperation Team of Florida's Disease Response Effort, the group that has convened this workshop today. Dana, over to you.
Excellent. Thank you so much, Emma, and good morning and afternoon, everybody. Thanks for joining us. I guess I'd also like to add that this webinar is one in a series that's been hosted by the Caribbean Cooperation Team of Florida's response to stony coral tissue loss disease, as well as AGRA and GCFI. And the NOAA Coral Reef Conservation Program, and the other webinars, which have focused on things like how to detect stony coral tissue loss disease, surveillance and monitoring methodologies, and intervention or treatment for stony coral tissue loss disease. Those recorded webinars are available on the AGRA Coral Disease Outbreak website, as well as the GCFI website for stony coral tissue loss disease. And we'll repost the links to those sites at the end because this webinar is being recorded and will be made available there as well. So I'm just going to quickly give you guys a broad idea of what we're going to cover in today's webinar. So you should be able to see the agenda on your screen scrolling down. And first, we're going to hear a little bit about what do we mean when we're talking about coral rescue in the context of stony coral tissue loss disease. What do we mean when we say rescuing corals from SCTLD? And we're going to spend a chunk of time learning about Florida's rescue effort as a part of their broader response to stony coral tissue loss disease. As everyone knows, this new disease was first observed in Florida, and Florida quickly identified rescuing healthy corals and saving them to support future restoration efforts as a really important piece of their response effort. So we'll hear all about that effort in Florida, and then we'll pause and have some time for questions and discussion with our speakers. And then we're going to hear about rescue efforts across the wider Caribbean region. other efforts underway to support the rescue of corals in areas that have been impacted by stony coral tissue loss disease outbreaks. And after those presentations, we'll have some more time for question and answers. And we know that many of you online today have either initiated efforts to support coral rescue as a part of your response to stony coral tissue loss disease, or you're thinking about it and starting to plan for it. So please, please share your plans and your ideas in the chat. Everyone, please, feel comfortable using the chat box. We have a Q&A box that we'll keep an eye on during the Q&A sessions after the presentations. But feel free to be sharing your ideas and reactions and responses or your efforts underway using the chat function. So with that, I think we will start with our first presentation, and I'm going to hand over the mic to my colleague at NOAA, Jennifer Moore. Jennifer works for NOAA Fisheries Southeast Regional Office in St. Petersburg, Florida. She coordinates the recovery program for threatened Caribbean corals protected by the U.S. Endangered Species Act. She's the co-lead for the Florida Coral Rescue and Propagation Team, and acting co-lead for the MPAConnect Reefs Coral Restoration Program in the Florida Keys National Marine Sanctuary. So Jen, feel free to unmute your mic and share your screen.
All right, thank you everybody for hanging in there with me. Hello, and thank you for inviting me to speak about this important topic. I'll be presenting an introduction to coral rescue generally, and then a case study of the Florida Coral Rescue Program. So what is coral rescue? It can be many different things depending on your situation, but generally the idea is that in response to a significant mortality, you collect corals or their gametes to ensure that you do not lose a species entirely in your location. What coral rescue looks like in your location really depends on several factors. First, you must identify your goal with your particular location and situation in mind. Perhaps you just want to collect small fragments for genetic preservation, or focus on extremely rare species. It'll be based on the status of your reefs in your particular location and the coral's response to the threats. Then you need to consider what resources you have or can obtain within the timeline that you can accomplish your goal. Think about the space and support you have for tanks, or if land-based rescue is not possible, what in-water resources do you have? Do you have existing coral nurseries that you can expand? Are they situated away from the reef to decrease the possibility of spread of the disease into your rescue nursery? In either case, the size of the coral you collect will dictate the size of the rescue facility that you need. And you will need to prioritize which species you want to focus on, taking your goal and resource assessment into consideration. You can always plan for unlimited resources, but make sure that it is scalable based on identified priorities. And then just do what you can. Coral rescue continues to evolve over time, and your project will too. Just because you may not be able to have a huge operation doesn't mean that you can't make a meaningful impact in response to a threat on your reef. If you don't have land-based resources yet, consider in-water rescue and maybe stage those corals for later transfer to land should those resources become available. But just try to do something, because doing nothing is not an option when we are faced with the type of destruction and mortality that an event like stony coral tissue loss disease is causing in Caribbean reefs. And so based on that framework, I'd like to present a case study of how we implemented this in Florida. To start off, I want to describe the risk, the situation that we were faced with in Florida back in 2018. Stony coral tissue loss disease is ravaging coral Caribbean reefs that I don't have to tell you guys about, and it's affecting over 20 species of stony corals. The disease was first discovered in Florida in 2014, and it spread up and down the Florida reef tract over the past 8 years, and has spread to many locations throughout the Caribbean. And due to its high rates of transmission, prevalence, and mortality, once a reef is affected, devastation is left in its wake. So now to discuss the response to this significant risk. In 2018, the state of Florida and US federal agencies took drastic measures to respond to the disease-induced mortality. And we came up with a plan to rescue corals from the wild, hold them in land-based aquaria to prevent them from being infected, retain their genetic diversity, and eventually reproduce them for future restoration. We initially started with presumably healthy colonies from ahead of the disease front, so we could reduce the risk of coral transmitting the disease in the aquariums. With over 20 species susceptible to disease, we needed to prioritize which species to rescue because we knew that it would be extremely challenging to focus on all of the species at the same time. We categorized the species into high, medium, and low priority based on things like susceptibility to the disease, rate of mortality, conservation status, and abundance of the population after affected by the disease. We developed a genetic management plan to guide rescue collections and ensure that sufficient genetic diversity would be collected to support restoration activities. The target was 50 unique individuals or genets per species because that captures approximately 90% of the genetic diversity of a population. But because we couldn't tell individuals apart in the field and genetic markers had not yet been developed for most of the species, we set a target of 200 individuals per species to be collected as geographically spread out as possible for a total of approximately 4,000 corals. We are developing genetic markers for all 20 species and have completed 10 so far, which will be made available to the community. Then we got down to the business of coral collections. In the fall of 2018, we piloted collections using dayboat operations, but due to the number of corals we were targeting to collect, the geography that we needed to cover, and the amount of post-processing to trim, mount, measure, photo, and tag the corals, we soon realized that a liveaboard operation would allow for maximization of effort and efficiency. Corals were carefully selected for collection, targeting 15 to 30 centimeter diameter colonies to balance them being large enough to hopefully be reproductive, but not exceeding a size that could be easily collected by a diver without assistance. We continue to refine our process and equipment to allow for collection of up to 400 corals in a single 6 to 8 day cruise. We conducted all of the post-collection processing on the vessel and kept the corals healthy and happy for the duration until they were delivered to an intermediate holding facility. In all, we conducted 7 cruises between 2019 and 2020. Due to the progression of the disease front west along the Florida reef tract, we conducted most of our collections in the Marquesas and the dry Tortugas. All of those corals were going to need lots of good homes. As soon as we realized that we were going to need to hold up to 4,000 corals and just how much space an individual coral actually needs to remain healthy, we reached out to the Association of Zoos and Aquariums as the only entity that had the suite of resources, expertise, and professionalism to take on this significant conservation challenge. The AZA established the Florida Reef Tract Rescue Project. My colleague Beth will be discussing the AZA partnership in greater details next. But in summary, we now have 26 facilities across 14 states that are currently holding rescue corals. We also have recruited 7 non-AZA facilities in 3 additional states to hold rescue corals. It is a truly unprecedented level of collaboration, and we could not be successful without all of these partners. But before the corals could be delivered to their long-term holding facility across the country, we needed to get all of those corals off the vessel and held for a period of time until the long-term facilities were ready to receive the corals. That is where Nova Southeastern University and the University of Miami stepped up to the challenge with large-scale holding space and expertise to care for the corals. It was from these two locations that corals were shipped all over the country to long-term care facilities. This network of facilities has grown the field of knowledge of holding Caribbean corals in human care exponentially. They are in constant communication about best practices, tips, and tricks, and support each other. Most of these species had not been held in human care, and definitely not in these quantities. That's not all that the AZA and the Coral Rescue Team has provided in terms of resources. Holding and caring for these number of corals in a way that will allow them or their offspring to ultimately be returned to the wild takes an insane amount of thoughtful consideration. As you can see, we have extensive guidance and amazing network of professionals volunteering their expertise to advise on all aspects of systems operations and coral care. And these resources are available to be shared with all who can benefit. And while retention of these corals to provide genetic preservation is a primary goal of coral rescue, there is a need to propagate these corals to assist in repopulating Florida's coral reef when the time is right. The Florida Aquarium has already successfully sexually reproduced 10 rescue species, both through lab-induced spawning with highly controlled conditions and in greenhouses with natural cues. We have recently prioritized 5 species for ramping up propagation efforts across several rescue facilities. These corals have been settled and reared for over a year or two in some cases. We have accomplished several firsts in the field of land-based propagation, including the first ever cross-fertilization of Diploria labyrinthiformis wild gametes with ex situ produced gametes, settled the larvae and reared them for approximately 6 months or so far. And just to hit home how novel and challenging this was, the wild corals were off Key Largo, Florida, and the rescue corals were in Apollo Beach, Florida, over 300 miles apart. We are using these rescue coral offspring to conduct research on best outplanting methods for these species, many of which have not been the focus of restoration to date. We are providing these corals to support other research and restoration efforts. And eventually, we want to complete the entire pipeline of land-based propagation to restoration using rescue corals, and have begun pilot projects and are supporting existing restoration plans. So what are the results? To date, we have collected over 2,300 corals from over 180 sites along Florida's coral reef, and are in a phase of only needing to conduct supplemental collections to round out our genetic diversity and to collect strategic survivors from the endemic zone to also serve as broodstock should stony coral tissue loss disease resistance or resilience be genetically based. We have developed a program that can be replicated in other locations, not only as a response to stony coral tissue loss disease, but for other mortality events that may have significant impact on coral populations. And now it's my pleasure to introduce my friend and partner in crime, Beth Furshow. Beth is the Florida Reef Track Rescue Project Coordinator with the Association of Zoos and Aquariums, and Beth is located in Virginia Beach, and the AZA is based in Silver Spring, Maryland. Take it away, Beth, and I hope you don't have any of the challenges that I did.
I'll cross my fingers, Jen. Thank you. All righty. Good. Come on, guys. There we go. Can you all hear me, I hope? All right.
Yep,
Ms. Beth,
loud and clear.
I'm from the Association of Zoos and Aquariums, and I am the project coordinator for the Florida Reef Track Rescue Project. I want to thank the workshop organizers for inviting me to be a part of today's workshop. My presentation is really a conservation story of hope for our backyard reefs here in the US. My story is focused on the Florida Reef Track, or also called the Florida's Coral Reef. But I hope that my story contains some inspiration, some facts that resonate with all of you as you go back to your campaigns to bring hope to your local reefs. My story begins with an invitation and a call to action. As Jennifer mentioned in her presentation, the state of Florida had an idea of removing thousands of corals from the reef track. It was part of a huge undertaking to make sure we had corals that would be the foundation of future restoration efforts and the future of Florida's coral reef. In 2018, an invitation went out to the Association of Zoos and Aquariums for reasons Jen mentioned. And really, the response from the association was very, very simple. And that was, when do we start? Before I get to the really meat of my storytelling today, it might be good to explain what AZA is and who AZA is. The Association of Zoos and Aquariums is a nonprofit organization dedicated to the advancement of zoos and aquariums. The AZA and its international 235 member facilities envision a world where, as a result of their work, all people respect, value, and conserve wildlife and wild places. Member institutions meet the highest standards in animal care and welfare and provide fun, safe, family-based experiences. Each year, member institutions collectively draw more than 200 million visitors to their gates and contribute millions of dollars to support scientific research, conservation, and education programs. Keep in mind that less than 10% of US Department of Agriculture permitted zoological facilities in the US operate and are accredited as AZA facilities. When it comes to animal care, welfare, conservation, and research, these facilities and the folks that work at them are the cream of the crop. And since 2018, AZA has been the Florida Reef Track Rescue Project. Within months of the FWC invitation to help with the rescue, the project was launched and recruiting of holding facilities began. And corals were moving into our country's heartland by April of 2019. In fact, our first facility to receive corals outside of Florida was a small museum focused on the Mississippi River in Iowa. And we've been busy ever since. Since 2018, our numbers have grown. Currently, our rescue network consists of 21 facilities with 22 partners managing those facilities in 14 states. And we're assisting the state with networking a number of non-AZA accredited facilities within the state of Florida and across the country. Supporting our holding facilities, there are 50 Friends facilities across the US. Now, these are AZA facilities that for whatever reason can't hold corals themselves, but want to be a part of the project. And they're providing funding, resources, and even manpower. And we can't forget our commercial partners, universities, colleges, and community colleges, and other government agencies that are also collaborating in the rescue network. And to measure the commitment of our AZA facilities to the coral rescue, you need only to look at the FWC rescue dashboard that Jen showed you earlier. Of the 2,341 rescued corals currently in holding, 1,650, or over 70%, are being cared for at AZA facilities. Over 50% of those corals rescued are being held outside of the state of Florida by facilities in other states. And to really hit home, the contributions and investment that AZA and its network of facilities have contributed direct to rescue, I'll share this slide. Of the estimated $18 million invested in coral rescue from all partners, AZA and its members have contributed over 62%, or more than 14 million to that effort from July 2018 to July 2021. Now that data is a bit outdated, I'll admit, but each year AZA facilities complete reports detailing the investment they have made in animal research and conservation efforts. The 2021 report should reveal that this investment is ongoing, and AZA's commitment to Florida coral rescue continues to be significant. So let's take a look at what the Florida Reef Track Rescue Project is and how it works. The project is a vast network of accredited facilities across the country supporting the care of Florida corals. Supporting those facilities are a number of working groups that drive the work of the project, and coordinating and supporting all that work is AZA, and my lucky opportunity to contribute to this effort as project coordinator. The project has always had a fourfold agenda: holding, enhancements, funding, and awareness. Since the start and now moving forward into our next 3 years, these priorities continue to be the same. So what do these goals, what does this strategy look like in action? Now in our 4th year, we're looking to recommit all of our holders to another 3 years of activity. The facilities listed here have already signed their commitment for the next 3 years. And all totaled, only one of the original holding facilities have resigned from the project. We have a full load ahead of us. We have 5 facilities currently exploring their involvement, and hopefully we'll be submitting requests for participation within the coming year. We also collaborate with 9 non-AZA universities and aquarium science centers that are also holding corals. So little by little, our network is growing, and it needs to, because in the next 3 years, the plan is to begin production of corals for outplanting back to the reef, as Jen mentioned. Some of the holding facilities will move into coral propagation activities to produce the corals needed. As Jen mentioned, the propagation plan is currently under development, and the genetic work being done by the state of Florida will be the foundation of ensuring the genetic diversity of the reef for generations to come. And the AZA is part of the planning for propagation. Even now, AZA facilities, as part of the CHOP working group, are advising the state on creating comprehensive protocols and procedures based on strong, modern zoological processes. The expertise and experience of AZA facilities will help ensure that the care and welfare of broodstock already in holding produces strong, healthy offspring for future restoration efforts. And AZA facilities like the Florida Aquarium, Mote Marine Laboratory and Aquarium, and the Florida Coral Rescue Center, which is a collaborative facility between SeaWorld and Disney, will help lead the propagation efforts, working with facilities across the country in large-scale production of juvenile corals for outplanting. Our second goal is enhancement. We realized early on in the project's development that our holding facilities could not do this alone. And we've always known that coral science entertained more questions than we have time or resources to answer. So we set out to support our holders, and along the way, if we could, we would work to answer some of those questions through our collaboration. Key to this was the incorporation of working groups. Working groups populated by holding facility representatives, community partners, and friends of the project were set up to provide arenas for collaboration, platforms for discussion, and knowledge building to help bolster our efforts for the long haul. To date, several working groups have been established and more are on the horizon. These working groups include the Holders Working Group that meets for an hour once a week, every week, just to talk about coral care and welfare. And the Coral Health Management Advisory Group that meets once a month formally. And from these working groups, products are developed that help our holders work smarter together. These products include the Water Quality Diagnostics Initiative and the Coral Health Diagnostics Initiative. With the help of our holders, we're also working on a coral aquarist training program that will build our next generation of coral aquarists. And we cannot forget our Equipment Resources Initiative that matches companies that supply key goods and services to the public aquarium community with our holding facilities. The initiative provides goods and services to our holders at greatly discounted prices. So I'll ask you to recall that earlier slide with that whopping $18 million number. In essence, that was startup and facilities finding spare this and extra that, recycling this and creatively resourcing that. That initial outlay is not sustainable over time. So finding ways to give back to our holders is critical. The enhancements I spoke of earlier, like the Equipment Resource Initiative, is part of that giving back, but more help is needed. Early on, we wanted to run this effort like a small nonprofit, making sure that we support our holders and we help them sustain their effort in exchange for the great care they were going to be giving these rescue corals. Funding support in that case was paramount. One of the key functions to date of the project is facilitating financial support to holding facilities. Directed, planned funding is important to the sustaining and to the establishment of any rescue or long-term nursery effort. For example, recent funding projections for one of our largest facilities in Orlando, the FCRC, home to over 700 corals for the next 3 years, is nearly $2 million US. The AZA is proud to have been part of this shepherding of state and federal funds to our holders over the last 3 years. From 2021 to 2022, the AZA has facilitated the fund distribution of nearly $850,000 in grant funding. And we want to take this opportunity to thank NIFWIF, FDEP, the Fish and Wildlife Foundation of Florida, and FWC for their generosity in supporting our holders. Communicating what we are and why we are doing it is critical on many fronts. We must make people aware that a national treasure is collapsing. We must communicate the need for support, and we must activate our citizens to use their voices in support of management policies that not only address the current crisis, but address wider coral conservation issues and hopefully make the need for what we're doing now not needed in the future. So awareness is our fourth priority. We've developed a web page on the AZA website that celebrates the project's accomplishments and acts as a communication hub for external audiences. The QR code that you've seen on this presentation throughout will lead you directly to our webpage. We're also using external platforms like lecture series, conferences, and webinars hosted by our member facilities and friends to share the story of rescue with others. In 2022, we're fundraising in hopes to launch a 12-part podcast series with the Speak Up for Blue network on the rescue and our holding facilities. We hope to fund our children's literature initiative that will take coral conservation and the coral rescue story to young audiences and be translated into the languages of the countries in the Caribbean who are also facing the loss of healthy reefs. And we're currently fundraising to launch a messaging campaign in collaboration with iHeartRadio to evaluate the effectiveness of coral conservation messaging so that we can ensure we're telling our stories in a way that encourages action as well as activism. So 8 years ago, these young kids were born. And now they know more about how to use a cell phone than I will ever know. But 25% of their lives have been spent in some sort of COVID response. And they've never known a time when Florida's coral reef or the Caribbean was not battling stony coral tissue loss disease. But at the start of this presentation, I said this was a story of hope for coral reefs. Working together with many partners, the AZA is making a difference for Florida's coral reef by safeguarding its species and supporting the restoration of the reef in the future, doing its part to make sure these kids can experience a healthy reef in their future. Bottom line, collaboration works. Working together here in Florida and reaching out to support you, our Caribbean colleagues, in your own battles for your reef survival, we can make a difference. It's important to remember that what we've accomplished in Florida is not the only way to get things done, not the only way to do things and be successful. Each of us have a variety of variables to address in making and executing plans to support the survival of coral reefs. What I hope you take from my presentation, my conservation story today, is this. I hope you feel empowered to continue to collaborate and to share. Is creative thinking, the thinking outside of that crazy little box, when looking about you for resources and the brain power and the stamina you're going to need to make a difference. We owe this to ourselves, to our reefs, and to our kids. Thanks a lot.
Excellent. Thank you so much, Beth. Okay, we have a few minutes for some questions for Beth and Jen, or some observations, comments. There's a Q&A box at the bottom of your screen. Everyone should be able to see the Q&A function. So feel free to submit them there, or we'll also be keeping an eye on the chat as well. Any questions or thoughts for Jen and Beth?
Dana, do you want me to start the ball rolling?
Sure. Thank you, Judy.
Okay, here's a question mostly for Jen, and she may not yet be able to answer it. She referred to the need for 50 unique genetic lineages in the corals that they were collecting that had not yet been exposed to SCTLD. And that was going to require about 200 corals because they didn't know their genotypes at the time of collection. And I was wondering if on the basis of the work that's been done to date, is there any revised estimate of how many different corals you should collect in order to get the required 50? Or what?
Yeah, so Judy, we were very pleasantly surprised that in the majority of the corals that we have already developed markers for and genotyped ours, which is about 10 of the species, and then actually doing the genotyping, there's not high levels of clonality. So that means that you probably don't need to collect 200 to make sure that you get 50 unique ones. And I can share all those details, about which species and how many, what rates of clonality and all that kind of stuff offline. But the good thing is you probably don't need to collect anywhere near 200 to get 50. But again, that's for, the certain species that we've already done, and it may be a different story for some of the other species. Most of those were broadcast spawners, so it is kind of intuitive that you might not have high levels of clonality close to each other, but with the brooders, you might have either more clonality or highly related individuals, and so you might want to spread out your collections more geographically. We can share the details though offline.
Okay, 2 more questions. Trip Funderburk wants to know if you found any coral genotypes that are resistant to SCTLD.
Well, that's not the purpose of our projects. I know there's a lot of research going on out there to look at the differences between individuals. Some clearly appear to be susceptible, while the one next to it might not be. So that's observations on the reef. And I know there's a bunch of researchers who are actually trying to look into the mechanisms of that. But I don't have the answer to that specifically in terms of like corals that we have in coral rescue that are or are not resistant to SCTLD.
And Katie Lesneski says, similar to Judy's question, how do you geographically constrain the populations in Florida?
Hey, Katie. So for our broadcast spawning species, we are in a very good situation that we have mixed populations, meaning the entire reef tract is one population. And so that's why we initially did all of our collections ahead of the disease front, because we could do that and be assured that we would still get a representative of the entire population. However, that's just based on genetics alone, genetic population structure, but we still wanted to diversify our collections in terms of habitat type and geography, because there might be different nuances there at play. And because we intend to use these as not only preservation, but also for reproduction and repopulation. So we did want to distribute our collections geographically, even though for the broadcast spawning species, it is one population. For the brooders, we are in the process of conducting a study to identify the population structure of the brooding species along the Florida reef tract. And so you have to stand by for the answer when we get the results of that study for the brooding species.
Okay, awesome. Well, if there are any other questions, or you guys think of any other questions for Beth and Jen, please put those in the Q&A or in the chat function. Jen and Beth, we would just ask you guys to please keep your eye on the chat or on the Q&A and go ahead and type your answers right in there. And we're going to move on and hear about some of the rescue efforts across the Caribbean region. So Emma, I'm going to hand the mic over to you to introduce our next speaker.
Okay, excellent. Well, moving from that amazing experience in Florida to the Caribbean region, I'd like to firstly introduce you to Logan Williams. She's based in St. Thomas in the US Virgin Islands, where she works as the Conservation Manager for Coral World Ocean and Reef Initiative, Inc., a newly established nonprofit located at Coral World Ocean Park. She's a member of the Virgin Islands Coral Disease Advisory Committee and she serves as the rescue and restoration team lead. So she's going to tell us about stony coral tissue loss disease and coral rescue in the US Virgin Islands, their past experiences and their future directions. Please go ahead, Logan.
All right. Hello, everybody. Thank you for having me with you guys. Just give me a second here. Can you guys all see the presentation mode?
Affirmative. Affirmative.
Okay, great. All right. So, yep, my name is Logan Williams, Conservation Manager for Coral World Ocean and Reef Initiative, Inc. And today I'm just going to, like you guys said, talk about our coral rescue operations here in the territory. So just a brief background. I am a part of the Virgin Islands Coral Disease Advisory Committee. And so as a member, we had included coral rescue in the original coral disease outbreak response plan, which was created in 2019, 2020. However, we really didn't have the resources and the funding, the personnel to conduct large scale restorations or scale this up in any way. But we were removing, and this was also, I should say, this was basically in the epidemic phase of this disease. So we were seeing massive mortality, especially affecting our smaller colonies, which were pretty abundant on some of our monitoring and intervention sites. And so we were actively removing these smaller colonies, basically 30 centimeters max diameter, from these affected reefs in an attempt to reduce the disease load within those coral communities. And these removed corals were transferred to UBI, where they were used for epidemiological research and treatment purposes, but also to test various treatment options. And some of them were also euthanized in a 20% bleach solution. And so we didn't really want to euthanize these corals. We preferred to try to rescue, treat, and rehabilitate them in our ex situ facilities. But like I said, we just didn't have the capacity to do that on a large scale at the time. But we did decide that we were going to, like I said earlier, start transporting these corals to UBI, where Dr. Marilyn Brandt was conducting some early treatment trials in her land-based facility. And some of those trials were effective, and these corals were treated. And so they remained within their land facility at UBI for temporary care. However, they wanted to increase the amount of space in their nursery to bring in more corals to basically further investigate the dynamics of this disease and how it was affecting these susceptible species. And so I had studied under Marilyn for my master's degree. And so at the time I was at Coral World, and she reached out to me and had asked if we would be able to develop a two-pronged coral rescue program between these two organizations, where Marilyn would continue to bring in these corals, they would be treated and then transferred to Coral World for permanent housing. And so coincidentally, at the time, we were actually renovating and rebuilding our indoor aquarium exhibit, which had been destroyed from Hurricanes Irma and Maria. And so we did have a tank that was open that we could place these corals in. And so this MOU was drafted between UBI and Coral World. And at the time, I was the education and research coordinator working for Coral World. I am now under Corey. But we basically drafted this 3-year commitment, and our responsibilities were to care for these corals, to fate track them, and then basically use these corals for educational gene banking and potentially future restoration purposes. These corals were housed in this indoor exhibit. It was an open system, but it did have an inline UV sterilizer connected to it to prevent pathogen introduction. But it was a natural reef aquarium, so there was some live rock and some sand, which was there to serve as a biological filtration mechanism. However, I learned basically at this time that since we were working with these quite fragile individuals, especially since they had undergone treatment we have since stopped using this aquarium and is in the process of being renovated. And our corals have now been transferred to our new land-based nursery, which I'll talk about a little later on. So like I said, we were removing these small colonies, but they were affected, they had disease. And so we needed to figure out a way to treat them before, being able to house them in our facilities. At the time, we did have the base to be amoxicillin treatment that we were using on affected corals in the field, but we wanted to conserve that supply because we didn't have a unlimited supply of base to be at the time. And so we basically use that for our larger corals to basically help protect them, prevent them from dying, basically, these larger reef building species that were susceptible and continue to become affected with this disease. And so we reached out to Cheryl Woodley and Carl Miller, and we had heard that they had experimented with ampicillin dosing. And so we didn't have ampicillin, but we did have an abundance of amoxicillin. And so we discussed that process with them. They basically explained their methods and protocols. And we decided that we were going to try this dosing method as a viable treatment option using amoxicillin. And so UVI conducted the controlled experiments, and they investigated the effectiveness of amoxicillin dosing treatments, which is 100 milligrams of powdered amoxicillin per 1 liter of seawater, sterilized seawater. And the coral is placed in that aquarium or container, what have you, for 10 days, but there are 100% water changes each day. And so this continues, like I said, for 10 days. And they compared this treatment method with other treatment intervention methods. We also at Coral World started doing the same thing. We started bringing in diseased corals and administering this daily dose for 10 days with 100% water changes, and either for 10 days or until all lesions were halted. And we found collectively that this can work, but it is much more successful if the lesion is amputated and then the coral is subjected immediately after amputation. This coral is subjected to these dosing treatments. And so the conclusion is that it is effective at treating and halting SCTLE lesions, but it increases with amputation. And since we were starting to bring in these corals and testing, these different treatment methods, we needed to create a database. And so we started to basically work on our coral rescue database, which is actually in the process of being updated right now. But that's kind of a screenshot of some of our metadata, some of the things we were recording. And these two images to the bottom, basically the one on the left is a Dendrogyra fragment that was brought to us and completed its treatment regimen. And you can see that lesion, that bright white area, was halted because of those treatments. And then the image to the right is a small Pseudodiploria strigosa fragment that was treated with the same methods at the University of the Virgin Islands and then transported to Coral World. So since we started kind of revving up our rescue operations, we found out that we could treat these corals, that we didn't want to start, we wanted to continue this process to save as much as we could basically at the time. And so we drafted some rescue protocols for our disease response plan in March of 2021. And like I said, this was to address the increase in coral rescue operations that were occurring in St. Thomas, but also in St. Croix. And a really exciting opportunity happened in the spring of 2021. Some fragments were removed from large diseased Dendrogyra cylindrus colonies in St. Croix, collected from various reefs in St. Croix. They were transported via seaplane using a semi-dry transport method to Coral World, where I received them and basically started to treat them with amoxicillin. And so to the right, these are some screenshots of our initial strike team rescue protocols. All right, so now a lot of things have changed since that time. Like I said, we ended up removing our corals from that indoor exhibit. We are now in basically construction phase. So we are in the process of constructing our land nursery and expanding it to take in more corals for various projects. And since we've started to increase these rescue efforts, at least a component of my job is to come up with a new updated coral rescue protocol. And this protocol would include our amoxicillin dosing methods. And this is a draft. I'm still working on it, but it should be finalized pretty soon. And then in December of 2021, we received 2 of our larger coral rescue. We engaged in 2 larger coral rescue operations here in the territory. First one, we received Dendrogyrus cylindrus fragments and small colonies that were collected from multiple reefs in St. John. And this was in collaboration with the Virgin Islands Park Service in St. John, and also CORE, which is a nonprofit foundation that does a lot of in-water intervention work. And we received a total of 11 Dendrogyrus cylindrus corals, and that included colonies and fragments. And these corals represented, well, we haven't done the genetic tests yet. We do have the supplies to do it, and it's something that we're going to try to do very soon. But we assume that we have 8 Dendrogyrus cylindrus genotypes total, 6 small colonies, and then 5 fragments that were collected from 2 separate colonies. Collection methods followed those that we recently drafted. And some of these corals were removed with active lesions. And so over time, I kind of figured out that, and we figured out as a group, that it's best just to remove that try to amputate that fragment, remove it without any lesions present. They're much easier to handle. And then you don't necessarily have to treat them with amoxicillin dosing. And just an update, all these corals are doing fantastic. We've had 100% survivorship. We have had a couple of cases where corals have shown potentially worrisome signs that could be indicative of SCTLD, but those areas were treated either using a small amount of base 2B amoxicillin, just a spot treatment, and then also using the amoxicillin dosing protocols. And so they were successful, and these corals are doing well. We also, basically the next day after receiving those pillar corals, we conducted our second seaplane transport from St. Croix to St. Thomas. And these corals were transported again using that moist or dry method. And so we received some Meandrina meandrites colonies and Dendrogyrus cylindrus colonies that were collected, or corals, I should say, that were collected from the Frederiksted Pier. Like I said, they were transported to Coral World. We did have a couple instances where we lost some of the Meandrina meandrites. Looking back on that now, I think it would have been beneficial to do a low dose amoxicillin treatment as a prophylaxis. But I was able to save the 1 colony through amputation and dosing, and it's doing great today. We also are now in the process of collecting corals for this large National Park Service restoration project. Basically, our job as Corey in this project, or this contract that we signed, is to collect and propagate and restore via outplanting 3 coral reefs in the Virgin Islands National Park. And some of this funding was used to expand and basically construct a new facility, expand that facility, add more water tables, stuff like that. And the project targets 7 coral species, 4 of which are susceptible. And those include Dendrogyrus cylindrus and then the 3 Orbicella species. And we started collecting corals for this project within the past month. So it's actually just started happening, which is very exciting. And our most recent coral census, and this includes microfragments, was about 742 fragments. And they represent, we think, about 20 genotypes. But included in this project, we have funds to conduct SNP genotyping. And so that will be very exciting. Like I said, we just started this, so, but I'm hoping to start doing this, collecting samples for this genotyping work very, very soon. Then we are also, this kind of got put on hold a little bit, this project, but it is still in the works. We did receive NIFWIF we were accepted, our NIFWIF project was accepted to basically really increase our treatment capabilities by constructing a land-based treatment facility and rehabilitation center that would treat corals brought in with SCTLD and then rehabilitate them. And these corals would remain in our care for permanent care, but also some fragments will be used for outplanting purposes in collaboration with the University of the Virgin Islands. And for this project, we are targeting 4 genotypes from the following species: Dendrodirus cylindrus, Copophilia natans, Montastera cavernosa, and Orbicella fabulata. And like
Logan,
I said,
1
yes.
minute, 1 minute warning.
Okay, so I'm wrapping up right now. Very briefly, like I said, we're doing a lot of stuff, but we are also in the process of updating our priority actions for restoration rescue since so much has occurred in the past year. And some of these priority actions are currently, we're working on them right now. And basically just stay tuned because things are about to really increase here in the Virgin Islands. And that's it for me. So if anybody has any questions, thank you again for having me.
Excellent. Thank you so much, Logan. Can I draw your attention, please, Logan, to the Q&A box? There are a couple of written questions there that you could go ahead and write back or reply to, and the participants can all see those as we go. And otherwise, everyone, please keep entering the questions there. I am pleased to introduce our next speaker, who is Margaret Miller. We're pleased to have you with us, Margaret. She is a coral field ecologist with expertise in population biology, reproduction, and disease ecology. She's the research director for SECOR International, based in Miami, Florida, and she's going to tell us about in situ approaches for larval propagation. Please go ahead.
Hi, can everybody see my presentation screen? I can't, it doesn't have the green
Yes.
frame around it, but very good. Good morning, good afternoon, everyone. I'm pleased to speak to you today. I'm going to have a little bit of a different slant than our previous speakers. I'm going to be emphasizing approaches related to coral larvae and specifically to approaches that can be implemented in situ. So you've heard a lot about very intensive land-based efforts. And there are some other possibilities that can be largely implemented in situ. Let's see if my slide will advance now. Guess it's a little bit sluggish. Okay, so I'm sure all of you are familiar with a very common now practice in the Caribbean in terms of propagating corals via fragmentation and outplanting, coral gardening approach, as it's known. This is a very effective way to produce more coral and grow coral biomass relatively quickly. It produces more coral in one sense, in terms of biomass, but in another sense, it doesn't produce more coral because we're cloning essentially the same coral that we started with. So the alternative approach is to use 2 separate genotypes to start with as parents, and those genomes can be recombined through their egg and sperm to form new genetic recombinations in coral larvae and settled coral juveniles. So this is the approach that I'm going to describe. It's more complicated, as we'll talk about, than fragmentation, but it provides the advantage of these new genetic recombinations that can provide more material for adaptation and natural selection to work on in the field. And we know this is important because all corals that we're placing back in the ocean are facing very rapid environmental change. So Corals have different species have different reproductive modes. The so-called brooders have a simpler mode where their larvae they have internal fertilization, and their larvae are released when they're pretty much already ready to settle. Most reef building species in the Caribbean are not brooders. More of them, and especially more of the SCTLD susceptible species, are these broadcast spawners. And so there are more steps involved because their gametes, their eggs and sperm, are released into the ocean. They have to undergo fertilization and all of these larval development phases in the water column. So that's a lot of steps, and we'll talk through some of those. Within the spawning species, there are also two types of species. The gonocores have separate male and female colonies. I've listed here some of the SCTLD susceptible gonocoric spawning species, and these can be addressed with larval propagation. The thing is, with the gonocores, it's much more challenging to collect large numbers of larvae. So folks have worked with these species, but I'm going to say that for large scale propagation and these techniques that I'm describing that are in situ based, the hermaphrodite species are much more amenable, and I've listed those SCTLD susceptible species here that we've done quite a lot of successful work with. So as I mentioned, there are many steps in this developmental the life history of these spawning corals. I will step through just examples for several of these steps, but I will emphasize, of course, that when those juvenile corals are being placed back out into the reef, they are subject to the same challenges that their parents were. And so this is still the persistent challenge that we have in this overall approach, is addressing the bottleneck that causes mortality to juveniles after they've been placed back on the reef. So the first step in this process is to identify the target species that you want to address, preferably within that list that I just showed you that can be successful with field gamete collection. But it's important to target your species because each species has many have a different or slightly different spawning schedule. The spawning is generally confined to a few nights during the year. But there are resources available from several groups that will regularly publish spawning prediction calendars for their regions. I show here just a small fraction of a spawning prediction calendar produced by our partners at Fundamar for the Dominican Republic. They're also produced by groups in different areas. So you can access these types of resources. The key points for each species are the number of nights after the full moon. So the corals are tracking a lunar calendar, as well as the time of sunset in your location. And then each species is also tracking the time either before or after sunset, in where they seem to be synchronizing their spawning. So these calendars, some of them can be accessed at the Coral Restoration Consortium website. And I can provide more information to access those resources later. But the collecting spawn is an exciting part of the process. Generally, these are night dives, so one needs to be very prepared once that predicted time is identified. We generally use some variation of this type of tent collector. Again, this is why the hermaphroditic species work better, because we can use this type of tent collector. You can see the eggs and the sperm are bundled together in these little bundles that you can see in this close-up of the collector cup. But I wanted to emphasize that spawn collection can also be synchronized with coral nursery operations, fragment nurseries. For the mounding corals, or most of the SCTLD susceptible species, it's a little bit more challenging because you need colonies of a certain size in order to be reproductive. But this can be integrated with field nursery operations. And then I also wanted to mention at least in Florida and perhaps in other heavily SCTLD affected areas, there is a strategy now to bring remnant colonies, sort of transplanting them into closer proximity in order to facilitate their natural fertilization potential. And so these so-called spawning hubs can also be specifically targeted, as their name implies, for collecting spawn. This is an example of a prototype of a larger spawn collector that we've been developing specifically for nurseries or these type of spawning hubs that can then collect spawn from many colonies at a time. The next step is fertilizing the eggs and sperm that are collected from different colonies. They do need to cross-fertilize, but I just want to emphasize a fancy laboratory or even indoor facilities are not required for this step. Here you see a group in Curacao performing that step. A small bit of equipment is required. A few wash bottles and a few pipettes are very helpful tools, but this can be accomplished on a boat or a beach or a parking lot, wherever you might be. There are then also options for taking those embryos that are fertilized and more or less depositing them directly into some sort of field-based mesocosm to complete their larval development and settlement phases, both. These images show the so-called cribs that C4 has developed and has, with our partners, been implementing in a lot of different areas in the Caribbean and some other areas. But you can see our primary strategy involves using artificial substrates. These are designed and generally made of either concrete or ceramic to settle the larvae, again, directly in this mesocosm. So there's no other handling that needs to take place during that stage. Then following settlement, those substrates can be outplanted directly onto the reef. Again, with no guarantees about their survivorship at that point, and various levels of husbandry effort can be applied to settlers which are settled in situ, either in nurseries or potentially bringing them back to a land-based facility at that point. And again, growing them up is a challenge, but they do grow up, at least some of them. I just want to mention quickly a somewhat alternative approach that we have piloted, that C4 has piloted in Curacao in collaboration with Peter Harrison, who has implemented this approach successfully both in Australia and in the Philippines. But we wanted to try it in Curacao. Generally, the benthic habitats in the Caribbean are probably of concern, we'll just say that. But in 2020, we did raise a large number of larvae. We used Coprophilia natans in this experiment, a large number of larvae in one of the cribs. But when they approached competency, instead of providing them substrates inside this mesocosm, we actually scooped them all back out again and filtered them down to a reasonable volume. And those competent larvae were applied directly to natural reef surfaces in these larval enclosures. And lo and behold, somewhat to my surprise, a year later, we were able to identify a number of Coprophilia natans recruits in these plots where we had done the active seeding. So this is just a cartoon that shows you a depiction of the different plots that we had either seeded or were controls. And so these are 14-month-old Coprophilias that had recruited in that direct seeding pilot study. One other tool that I wanted to mention, these are again a product of the Coral Restoration Consortium. C4 has contributed a lot to this effort, and there's a lot of effort that goes into them. So I think there's about 3 or 4 species that are available now, but an intention to expand the library of these so-called coral species fact sheets, which simply compile all the known information regarding larval propagation for each species. And so this includes sort of hints and lots of photographs and things and the spawning prediction times for each species. So these are also available on the Coral Restoration Consortium website. So I just wanted to sort of show you, SECOR works with local partners doing restoration in local locations, and you're going to hear about a couple of those local efforts and some of their accomplishments in some of the following talks. But just to show you that these practices are underway in many sites throughout the Caribbean, including, I've highlighted here several of them that have been highly affected by SCTLD, and some of our other partner locations, whereas there's been perhaps less, but certainly presence of SCTLD in the regions that those partners are operating. Very good, that completes my talk. Thank you. And I'm happy to address questions, or as I said, several of the following talks I think are going to relate to some similar methods. So I'm not sure how you want to do the questions.
Okay, fantastic. Thank you so much for that great presentation, Margaret. We will take some questions after the next presenters, but please keep monitoring our presenters. Logan, Margaret, please keep monitoring the Q&A box. There are a couple of questions there, and we'll come back to one or two live ones at the end. For now, I would like to introduce to you all Andreina Valdez, who is the Biodiversity Program Coordinator at the Foundation for Marine Studies in the Dominican Republic, Fundamar. She's going to tell us about coral rescue through larval propagation in the southeastern Dominican Republic. Thanks, Andreina. Can you go ahead and share?
Thank you, Dana. Margaret, can you stop sharing, please?
Okay.
Thank you.
Okay, ahora vamos a español, ¿no?
Sí. Buenas tardes, mi nombre es Andreina Valdez y soy la coordinadora del Departamento de Biodiversidad Marina de la Fundación Dominicana de Estudios Marinos. Hoy les voy a presentar un breve nuestros trabajos, esfuerzos de recuperación de especies susceptibles a Stony Coral Tissue Loss Disease a través de la propagación larval en el sureste de la República Dominicana. Antes, quiero actualizarlos brevemente sobre el estado de Stony Coral Tissue Loss Disease en toda la República Dominicana y presentarles otros esfuerzos importantes en el país para el monitoreo de la enfermedad y la recuperación de especies susceptibles. Gracias a esfuerzos de muestreo de enfermedades a nivel nacional, en coordinación con la Red Arrecifal Dominicana, de Nature Conservancy y el Ministerio de Medio Ambiente, sabemos que se ha vuelto endémica en la costa noreste de la República Dominicana, con prevalencia por debajo de un 10%, y episótica en el noreste con prevalencia promedio entre 29 y 32%, y ausente en otros sitios de la República Dominicana, aunque recientemente hemos observado la posibilidad de que haya llegado Stony Coral Tissue Loss Disease al sureste. Por su parte, el programa de restauración de arrecifes de coral de Wave of Change de Iberostar en República Dominicana, además de participar en el Plan Nacional previamente mencionado, cuenta con instalaciones en tierra capaces de salvaguardar fragmentos y colonias de especies susceptibles, que funcione como banco genético. Ahora de regreso con los esfuerzos de Fundemar en el sureste de la República Dominicana. Fundemar se ha dedicado a la conservación de los ecosistemas y recursos costeros marinos desde hace más de 10 años a través de dos programas principales, el Programa de Protección de Mamíferos Marinos y el Programa de Biodiversidad Marina, el cual está centrado en actividades de conservación de arrecifes de corales, como es la jardinería de corales con Acropora palmata y Acropora cervicornis, y la reproducción sexual asistida, de la cual hablaremos más a fondo. Debido a sus beneficios potenciales, ayudar a la reproducción sexual de los corales se ha convertido en un pilar de las actividades de restauración de corales, no solo en forma rentable y factible de producir un gran número de corales, sino que también puede aumentar la supervivencia durante los cuellos de botella, en etapas tempranas de la vida, y por lo tanto puede promover una mayor diversidad genética en las poblaciones de coral amenazadas, dándole las herramientas genéticas necesarias para adaptarse a un entorno rápido y cambiante y ante enfermedades emergentes como es el Stony Tissue Loss Disease. Fundemar es la primera institución en implementar estas técnicas en la República Dominicana y hasta ahora es la única, y establecimos la primera instalación de cultivo de larvas en el país. Hoy les compartimos 3 componentes que fueron clave para nosotros incorporar estas técnicas como un programa permanente en nuestra institución. Primeramente, la existencia de un programa permanente y robusto de restauración de corales permitió la exploración de técnicas novedosas con alianzas con organizaciones tanto locales como internacionales, creando mecanismos autosostenibles basados en nuestro contexto local para no depender completamente en ganar grants. En nuestro caso, ofrecer una experiencia de conservación marina a programas de voluntariado internacional proporcionó este mecanismo. En Valladolid creamos una estructura beneficiosa tanto para la industria turística local como para Fundemar a través de la adopción de viveros de corales. Las autoridades locales apoyan nuestro trabajo a través de permisos y las organizaciones privadas brindan un valioso apoyo en especie a nuestras actividades marinas, como barcos, equipos de buceos, tanques y tiempo de personal. Esta estructura robusta nos permitió invertir tiempo y recursos en explorar técnicas novedosas que podrían integrarse en nuestro programa en alianza con organizaciones internacionales. 2 socios internacionales fueron esenciales para implementar estas técnicas, como son Seacon International y The Nature Conservancy. Con su apoyo, capacitamos a nuestro personal durante talleres en México y Curazao y recibimos los conocimientos básicos para replicar estas técnicas en casa. Implementamos pruebas piloto en 2015 y 2016 con equipos muy rudimentarios y su éxito nos motivó a ampliar los esfuerzos. En el 2017 y 2018 nos centramos en capacitar al personal y recopilar la mayor cantidad de datos posibles sobre el tiempo de desove de nuestros corales locales. A través de la alianza con Seacon pudimos proporcionar, tener con nosotros las innovaciones de las CRIPs para cultivar fácilmente grandes cantidades de larvas in situ. El laboratorio de Corallium en México nos mostró un ejemplo de una instalación terrestre que sea rentable y a partir de entonces comenzamos a pensar en las posibilidades para nuestro entorno local. Entendiendo la importancia de tener una instalación terrestre para proyectos de investigación y cómo salvaguardar en caso de tormentas y huracanes, dado que nuestro espacio de oficinas ya es bastante pequeño y que las instalaciones debían estar cerca de la costa para el bombeo de agua, pensamos que una instalación móvil dentro de un contenedor sería más adaptable. Los desafíos fueron el diseñar un laboratorio específico para la cría de larvas de coral y encontrar subvenciones para financiar la construcción, lo que pudimos hacer gracias a nuestras alianzas con socios internacionales. Aquí presentamos una descripción gráfica del diseño de nuestro laboratorio. El laboratorio móvil se colocó frente a la costa para bombear fácilmente el agua del mar al depósito del laboratorio, que se encuentra marcado con la flecha naranja, donde se filtra y esteriliza antes de entrar a los acuarios. Tenemos 4 sistemas de acuario. En el diseño solo se muestran 3 porque es un diseño viejo. Y cada uno de estos sistemas cuenta con 3 tanques con su propio sistema de circulación. El laboratorio es capaz de contener más de 1000 sustratos de asentamiento. Integramos un sistema que automatiza el trabajo de mantenimiento, tanto como sea posible, como un sistema de plomería para rellenar y drenar tanques, sistema de flujo de agua y de filtrado para acuarios, enfriadores y calentadores para controlar la temperatura del agua y actualizaciones en vivo de la temperatura del agua. Dado que este es un campo de estudio bastante reciente, no queríamos solo replicar técnicas, sino también contribuir al conocimiento científico sobre el tema. Un ejemplo es que cultivamos exitosamente reclutas del coral Dendrogyra cylindrus en vivero in situ, el que se ve en la imagen de fondo, algo que no se había reportado anteriormente. También adoptamos un marco de diseño experimental y protocolos de monitoreo estandarizados. En primer lugar, formulamos claramente hipótesis para el experimento de restauración basados en la comparación de variables medidas en sitios experimentales de referencia y en nuestro control para estimar eficazmente el éxito de la restauración. En general, nuestro objetivo es mejorar la supervivencia a corto y largo plazo de los reclutas de coral. Entre 2019 al 2021, utilizando el laboratorio y los scripts de SICOR, producimos con éxito reclutas sexuales de 5 especies: Diploria laberyntiformis, Dendrogyra cylindrus, Acropora palmata, Acropora cervicornis y Colpophila natans. En total, para estos años, producimos más de 12.000 unidades con asentamiento, con aproximadamente 500.000 reclutas sexuales de coral que fueron sembrados en un área del arrecife, totalizando alrededor de 3.000 metros cuadrados. Queríamos también compartirles fotos recientes de reclutas de distintas especies. Aquí tenemos ejemplo de un recluta de Diploria laberyntiformis de 3 años, Diploria laberyntiformis de 2 años, Colpophila natans de 1 año y medio y ejemplos de Dendrogyra cylindrus de 1 año y medio crecidos en vivero in situ. Es importante resaltar que en los sustratos con reclutas que sembramos directamente al arrecife, como se hace con las otras especies de coral, no se han encontrado sobrevivientes. En cambio, los sustratos que fueron colgados en estructuras de vivero son los que han sobrevivido y crecido rápidamente, contando con más de 11 reclutas, dando soporte a este manejo como un método efectivo de cultivo de reclutas de estas especies. Esperamos que estas técnicas puedan continuar expandiéndose y replicándose en otros lugares del Caribe para que podamos dar colectivamente un futuro a nuestros corales. Gracias por la atención y quedamos atentos a preguntas.
Okay, muchas gracias, Andreina. There is some very good practical advice there between Logan
Welcome.
and Andreina. So let's now move to our next presenters, our next country. We have a fabulous threesome from Mexico who are going to tell us all about their work on biobanking and biorepositories to rescue stony corals. Our 3 presenters are Melina Soto, who is the Mexico Country Coordinator for the Healthy Reefs for Healthy People Initiative. Celia Romero is with the Mexican Fisheries Institute in Pesca. She collaborates with Dr. Claudia Padilla in coral restoration, and she focuses on the care of corals. And Anastasia Banaszak, who is with the National Autonomous University of Mexico. Her research focuses on developing low-cost techniques for scaling up larval-based restoration and innovative methods to rescue coral species. So, with that, Melina, can I pass this to you?
Muchísimas gracias, Emma, y muchísimas gracias por la invitación. Y bueno, pues arrancamos nuestra presentación sobre nuestro proyecto de rescate de corales usando biobancos y biorepositorios en el Caribe mexicano. Como antecedente, les recuerdo que la enfermedad de pérdida de tejido empezó en México en el 2018, pero rápidamente se propagó a lo largo de los 400 kilómetros lineales de nuestra costa, y en menos de un año habíamos perdido el equivalente a lo que veníamos perdiendo en los últimos 40 años por el cambio climático y otras amenazas. Entonces se pueden dar cuenta que la velocidad y la tasa de mortalidad que experimentamos era sin precedente y generó un estado de emergencia. Por eso, muchísimos socios o aliados, digamos, de los arrecifes de la parte del Caribe mexicano se unieron para hacer un plan de acción del síndrome blanco. Este plan de acción tiene 5 líneas estratégicas, de las cuales se derivaron en 57 acciones y fueron un poco más de 120 participantes para generar este plan de acción de todos los sectores de actividad, desde gobierno, academia, empresa privada, y obviamente organizaciones de la sociedad civil. Y una de estas acciones dentro de la línea estratégica de investigación y academia era justamente el diseñar un proyecto de rescate de material genético, un banco genético salvando material vivo, o sea, tejido, pero también criopreservando gametos. Entonces, pues nos unimos todas estas organizaciones y cabe destacar que pues unimos esfuerzo de manera intersectorial. Tenemos a la UNAM, una academia, ¿no? El Centro de Investigación, Universidad, pero también agencias de gobierno, organizaciones de la sociedad civil y también sector privado con el acuario del grupo Xcaret. Entonces desarrollamos este proyecto, diseñamos este proyecto que tiene pues tres principales objetivos, digamos. Es obviamente el tema de hoy, es el rescate de fragmentos o colonias sanas para resguardarlas en cautiverio, pero también es el atender el desove de estas especies susceptibles al síndrome blanco, enfermedad de pérdida de tejido, tanto en el laboratorio como en el arrecife. Y gracias a estos gametos colectados vamos a poder generar reclutas de coral o criopreservar también estos gametos para pues justamente prepararnos para el futuro y ojalá diseñar proyectos de restauración. Entonces pues empezamos con obviamente la difusión del proyecto con este tipo de banners de Se Buscan, porque desafortunadamente, como les comenté al principio, la enfermedad nos pegó muy duro, muy rápidamente y se volvía muy complicado encontrar estas colonias que queríamos rescatar. Nos enfocamos en las colonias que están altamente susceptibles a la enfermedad y difundimos eso con el público en general, pero también con un enfoque hacia los usuarios de las áreas marinas protegidas y en particular con, por ejemplo, las tiendas de buceo, los guías, etcétera, para que nos pudieran compartir la ubicación de estos corales. Y bueno, pues una vez ubicados, se rescataron. Adelante, Arce.
Muchas gracias, Meli. Hola, ¿cómo están todos? Bueno, yo les voy a platicar un poco sobre la colaboración que ha tenido el INAPESCA para este proyecto. Estos banners de los que nos platicaba Melina tuvieron su efecto. Por supuesto, las personas tienen muchas ganas de colaborar en el cuidado de nuestro arrecife, por lo que el llamado fue atendido, ¿no? Entonces recibimos muchas reportes de colonias de las más susceptibles a esta enfermedad. En total, nosotros en el INAPESCA contamos con 83 colonias diferentes de 62 genotipos, de las cuales 14 son de las 14 de las especies más susceptibles de los diferentes 17 sitios diferentes en México. Bueno, nuestros sistemas están divididos en dos grandes partes que son adecuados a las diferentes condiciones en las que se colectan las colonias. Uno se basa en dos sistemas de mesocosmos que cada uno tiene una capacidad de 35,000 litros y que mantiene condiciones semicontroladas. Aquí estos reservorios se abastecen de agua filtrada y una malla sombra que los protege de la luz directa del sol. En estos mesocosmos se mantienen colonias que son sanas, que son colectadas del medio natural, pero son sanas y que muchas de ellas mantienen una talla reproductiva. Otro gran sistema que tenemos en nuestras instalaciones son los sistemas de cuarentena. Estos sistemas son de condiciones controladas. Aquí mantenemos la temperatura del agua a control también mantenemos la calidad del agua con sistemas de filtración y sistemas de UV. Además de que, bueno, esos sistemas fungen como enfermería en nuestras instalaciones. Aquí llegan las colonias que han sido reportadas enfermas y aquí las atendemos. Igualmente se han mantenido con protocolos de tratamientos, muchos de ellos también con amoxicilina. Y bueno, también mantenemos aquí colonias de las especies más susceptibles, como son Dendrogira cylindrus y Meandrina meandrites. Cabe destacar que de todas estas colonias que hemos rescatado, el porcentaje de sobrevivencia ha sido del 80%. Siguiente, Meli, por favor. Bueno, y todo esto no queda solamente en el rescate del tejido vivo de las colonias, sino que tratamos de adecuar estos sistemas también para reproducir los corales. Nosotros mantenemos en nuestras instalaciones 29 colonias de talla reproductora, de las cuales 10 han manifestado algún comportamiento reproductivo. 2 de ellas han sido Dendrogira cylindrus, que resultaron ser machos. También tuvimos el desove de Diplorias labyrintiformis, de Orbicelas annularis, de Montastrea cavernosa, de Pseudodiploria estrigosa. En todo caso, de que no se pueda realizar la fertilización porque no hayan desovado 2 colonias de la misma especie, también colaboramos mucho con la parte de la criopreservación. Siguiente, por favor. Gracias a nuestros compañeros de la academia, el equipo de la UNAM, podemos nosotros monitorear todos los años estas colonias de talla reproductiva y en caso de que haya desove, nosotros podemos guardar esta muestra y llevarla a sus instalaciones para que se haga este método de criopreservación. En total, hemos ayudado a la criopreservación de el esperma de Dendrogira cylindrus y de Pseudodiploria estrigosa. Entonces, de este tema de criopreservación nos habla nuestra experta Ania. Muchas gracias.
Gracias, Arcelia. También se estableció un biobanco para colonias de las especies afectadas por esta enfermedad en el sistema de acuarios del Grupo Xcaret. Las colonias recolectadas fueron resguardadas en cuarentena y luego en mesocosmos. En total se resguardaron 52 colonias de 10 especies provenientes de 7 sitios. En este sistema de acuarios también están manteniendo reclutas sexuales que producimos en la UNAM y se está dando el cuidado y el mantenimiento de estas reclutas en colaboración con mi equipo. La siguiente, por favor, Meli. Gracias. En la UNAM hemos enfocado en producir nuevos corales de especies susceptibles a esta enfermedad en colaboración con Seacore International. Hemos trabajado con 4 especies susceptibles. Hasta la fecha son Orbicella faviolata y annularis, así como Diploria labyrintiformis y Pseudodiploria strigosa. Hasta la fecha hemos sembrado aproximadamente 350 reclutas en arrecifes de Puerto Morelos y pronto estaremos sembrando más de estas especies. Además, algunas de estas reclutas, por ejemplo de Pseudodiploria strigosa, fueron producidas al cruzar una colonia enferma con una colonia sana. Los reclutas ya de este cruce están sembrados en el arrecife y creciendo bien sin ningún signo de esta enfermedad. La siguiente, por favor. Por otro lado, como comentó Arcelia, hemos formado el Biorepositorio Mexicano de Gametos de Coral. En este caso, el enfoque es en las espermas que se preservan y se congelan a temperaturas muy bajas, ya que se pueden mantener así por tiempos muy largos. Hemos logrado producir criobebés o colonias de corales producidas por el cruce de óvulos frescos con espermas congeladas. Un ejemplo de la importancia de este esfuerzo es que en el biorepositorio contamos con espermas de colonias de Diploria labyrinthiformis que ya han muerto. Entonces esperamos que en el futuro podemos usar estos espermas para cruzar con óvulos para producir nuevas colonias. Como pueden ver en la diapositiva, estuvimos dando seguimiento a 9 diferentes colonias. Todas estas ya tenemos los espermas en el biorepositorio, pero 7 de estas ya han muerto. Entonces, lo único que queda de ellos son estas espermas congeladas. Gracias.
Gracias, Anya. Y como otros productos pues, de difusión que hemos realizado con el proyecto, hemos realizado diferentes infografías que se comparten en las redes sociales de todas estas organizaciones y también hemos organizado un webinar con pues, la mayoría de las presentes en este taller de hoy en día. Entonces lo pueden volver a ver en la página YouTube de Healthy Reefs. Cabe destacar que en realidad este proyecto que cuenta con una parte de financiamiento de Marfan también cuenta con en realidad muchísimo aporte en especie, digamos, de las diferentes organizaciones, ¿no? Toda la infraestructura, etcétera, forman parte de sus programas anuales de estas organizaciones y cuenta con el apoyo y el compromiso de todas estas personas que ven en la pantalla, que son capacitados y súper dedicados a justamente llevar a cabo el proyecto y llevar los objetivos a su éxito, y para eso pues se dedican muchísimas horas de su vida profesional, pero también personal. Entonces pues otra vez un gran agradecimiento a todas las personas capacitadas y comprometidas con este proyecto. Y muchísimas gracias y esperamos sus preguntas.
Excellent. Muchas gracias a nuestros compañeros de México.
Oh,
Thank
Emma,
you,
we can't hear you.
Oh. Hello.
I can hear her.
I can
There.
hear you.
Oh, okay.
Okay.
She sounded very, very faint to me.
Okay. Thanks,
Okay.
everyone. Thank you so much. That's the excellent presentations, and I think that starts to give us a feel for the different options and different approaches to coral rescue that are happening in Florida and also now in the Caribbean region. Let's have a look then for some of our question and answer section. We've had a couple of questions come in. I noticed there was one that you flagged, Logan, that you would like to answer live. Has been answered live. Was there another one?
I think it was the one about whether or not the rescue plan would be available, and it looks like she provided an answer saying yes.
She has. There you go.
And Logan, we're happy to distribute that for you too.
I think I accidentally clicked the live button, but
okay. No problem. So there's some really interesting protocols and materials and guidance that have been developed by the Aquarium and Zoo Association that Beth mentioned. Logan has mentioned that protocol, that new information, new guidance that's coming out of the Caribbean. We had some questions related to costs, and thank you, Logan. I see you answered that as well. I wonder, I know Anya, Melina, Celia, your team, that's something that you've tried to venture into coral rescue work on a less costly basis. would you have anything that you would add to the question that we received from Department of Environment in Cayman regarding costs?
Bueno, contesto una parte y después Anya. Por ejemplo, los 2 años de este proyecto, para empezar ese proyecto piloto, entonces como la base, digamos, con el cual hemos empezado, fueron 2 becas de Marfan, 2 grants de Marfan de $20,000, 20 y pico mil dólares. Entonces fue muy poco y por eso los laboratorios, tanto de la doctora Claudia como de la doctora Aña, tuvieron que poner contrapartida para poder cubrir los gastos corrientes, ¿no? del proyecto. No sé si quieres indagar un poco más en ese tema, Aña.
Sí, muchas gracias, Melina. Bueno, eso ha sido como un enfoque de mi laboratorio, buscando técnicas de bajo costo. Y yo creo que una de las cosas es ser muy creativo y siempre estar buscando cómo minimizar costos y colaboraciones. Yo creo que esto apoya mucho, tener colaboraciones y así tenemos más ideas y más posibilidades de reducir costos. Por ejemplo, en nuestro caso, los cribs que mostró Margaret, desafortunadamente en nuestra situación no funcionan muy bien, especialmente por las arribazones de sargazo o sargassum. Entonces, nosotros usamos una técnica distinta, un poco como tienen en Fundemar, como mostró Andreina, pero lo hemos adaptado a nuestra situación y creo que tenemos un sistema en tierra en donde podemos manejar diferentes especies al mismo tiempo y a un costo muy bajo, o sea, yo diría menos de $5,000 tenemos una estructura que podemos manejar aproximadamente 1 millón de embriones. Entonces, tenemos varios diferentes diseños y tenemos unas publicaciones sobre esto que esperamos que podemos sacar pronto. También para criopreservación hemos desarrollado un sistema que podemos llevar a diferentes sitios, al igual que estos laboratorios. Los hemos llevado inclusive a Belice y a distintos sitios remotos en México a muy bajo costo. Por ejemplo, establecer un sistema por menos de $500. Y entonces si alguien quiere información, con mucho gusto lo podemos compartir. Y también hemos desarrollado el sistema para criopreservación que podemos llevar a distintos sitios también. Entonces, creatividad creo que es la clave.
Excellent. Okay, thank you very much. There were a couple of questions that came up in the Q&A that might be worth just revisiting to make sure everyone saw those. There was one that Turks and Caicos Reef Fund asked, which Jennifer Moore kindly answered. And this is an interesting one. In the Turks and Caicos, they commented that they're not finding more than just a handful of remaining corals like Meandrites and Dendrogyras. So is there merit in collecting fragments for future restoration? Realizing that they may not get as many genotypes. Jennifer, you commented on that one. Are you still with us? Would you like to add any comment, if so?
Yeah, I'm still here. I mean, there's absolutely merit because with those species who are particularly susceptible, if you don't collect them, there's a very high likelihood that they'll die anyway. So that's the first thing, is preservation. So you won't have them surviving if they die of the disease out on the reef. So definitely collect them. And then, as I shared in the chat, based on doing more investigation into population structure, it may be appropriate and viable to cross reproduce your local corals with corals from other closely related or closely distanced locales, that you can then use those for repopulating your reef. You basically need to just take best practices for genetic management into consideration when doing reproduction, so that you don't inadvertently create bottlenecks by reproducing a very small population and putting them back out on the reef. So it's absolutely imperative that you do the rescue so that you actually have the material for the future. But it may not be appropriate to only repopulate your reefs with those few colonies that you're able to collect.
Okay. Thank you very much. Okay, that's good advice. Now, Judy Lang from AGRA, I believe. Judy, you've got a question. If you'd like to unmute yourself.
Yes, can you hear me now?
Yes, affirmative.
Good. I had a question focused initially on the Mexicans, but really anybody can also answer it. And that is that if initially you have to choose between saving whole colonies or fragments of whole colonies that either are naive to SCTLD, like the Floridians did, or have survived so far, which is more typical of the Caribbean, versus choosing to enhance the creation of new genetic diversity with the assisted larval propagation as advocated by Secor's approach, and maybe also implicitly promoted by Anya in her answer to the costs involved of doing that process. Which approach would you start with? You're a small island with a small budget. How do you know how to begin?
Well, yeah, I'll have a stab, and I'm sure that at the answering this question, I'm sure others will want to put in their opinion as well. That's a really tough question. I think it all depends on what the situation is. I think that there's no